right, so guys, I'm going to go through all the different C words. So in interphase, during the S phase of interphase, all of the DNA has to unwind and unzip so that DNA replication can happen. And at that point, those long strings of DNA are considered chromatin. Okay, so those long strings of DNA are chromatin. Okay, then in prophase, when the cells, or sorry, the chromatin begins to condense, what's going to happen is that chromatin is going to wind up, okay, and that's where we get that condensed chromosome. Okay, so we're going to represent that with one of these markers. Okay, so chromatin, okay, long string. That's going to be an interphase because that's when this S phase specifically, because that's when DNA replication happens. The DNA has to unwind and unzip so that it can then undergo DNA replication. Okay. Then again, when all of that winds up really tight, we have our condensed chromosome. Okay, so that's what each of these markers are going to represent, is a condensed chromosome. All right, so here we have a chromosome, okay, which is also one chromatid. And remember, we've got specific DNA on here. So let's say this is from the mom and this is from the dad. Okay, so we've got chromosome 1, because let's say that the genes that we're looking at are right here. Okay, and the reason why they are homologous chromosomes is because they do contain the same genes that then code for the same trait. So it takes two, one from mom, one from dad, to code for that same trait. So these two right here, okay, are homologous chromosomes. So here we've got one chromosome and one chromatid. Okay, this is before DNA replication happens. Here we've got another chromosome and another chromatid, so one chromosome, one chromatid. But collectively here we've got two different chromosomes, but because they have the same gene that codes with the same trait, that means that those are homologous chromosomes. It doesn't matter if they are not duplicated, or here is after DNA replication has happened. Okay, so here DNA replication happens. So again, this is, let's say this is from dad. So this was the original DNA. DNA replication happens, so this should be the exact same copy as this one because DNA replication happened. Okay, so exact same copy from dad. So two copies of the exact same DNA, which means these two are sister chromatids held together by a centromere. Okay, so here we've got, from the mom, we've got one whole chromosome and sister chromatids held together by a centromere. Okay, because these two, so one from mom, one from dad, have the same genes, the same traits that code for the same trait here. That makes these two homologous chromosomes. Okay, now the smaller, sorry, I couldn't find the exact same highlighter for the pink one. Okay, so now we have, let's just call this chromosome pair number 2. So that first one's, the bigger ones were 1, these are 2. Okay, so again, we've got chromosome from mom, chromosome from dad that contain different genes. So different genes that code for different traits. So here we have one chromosome, two chromatids. So those are your sister chromatids. A different chromosome with different chromatids. So again, sister chromatids here. But because here and here, when they come together later on, that's going to make a trait. So these right here are going to be your homologous chromosomes. Okay. Homologous chromosomes because they have the same traits. So again, these are held together by your centromeres. One came from dad, one came from mom. Same thing here. Homologous chromosomes because one came from mom, one came from dad. But individually, this is one chromosome, two chromatids because DNA replication has happened. One chromosome, one chromatid because this is like either after cell division has happened or before DNA replication has happened. So one chromosome, one chromatid. one chromosome, two chromatids, otherwise known as your sister chromatids. All right, so all of the DNA replication, okay, chromosome, replication, sister chromatids, all that DNA replication happens in the S phase of interphase, okay, and then they begin to condense in prophase, and then they line up in the middle in metaphase. Okay, so again, what we're going to end up having here is all of these. Remember, these just represent 4 of your total 46 chromosomes. Okay, so at this point, okay, these are still homologous chromosomes. These are still homologous chromosomes. Okay, we've got sister chromatids, sister chromatids, sister chromatids, sister chromatids. Now, once we start with anaphase going into telophase, and these begin to separate into 2 new cells, okay, so now these 2 green ones are now sister chromosomes because they're now going into 2 separate cells. These 2 are sister chromosomes, sister chromosomes, sister chromosomes. Okay, but these 2 here are still going to be homologous chromosomes because they have the same genes and the same traits. These 2 are still homologous chromosomes, same genes, same traits. Okay, I'm going to use the string again to divide my cells. Okay, so now I've got 2 different cells here. All right, so in this cell here, we've got those 2, okay? Homologous chromosomes, homologous chromosomes. Okay, over here, we've got our other cell. I didn't get 2 strings, I'm sorry. So now we have this string over here. All right, so we still have our homologous chromosomes, homologous chromosomes. But individually, this is 1 chromosome, 1 chromatid, 1 chromosome, 1 chromatid. They're homologous because they have the same genes and the same traits on them. 1 from mom, 1 from dad. Individually, this is 1 chromosome, 1 chromatid, 1 chromosome, 1 chromatid. Together, they're homologous chromosomes because they have the same genes and the same traits from 1 from dad, 1 from mom. I hope that's clear.